Hello everyone, I am Doyel Pandey. I am an assistant professor in the School of Mathematical and Statistical Sciences at IIT Mandi. It is my pleasure to present my research project titled Mathematical Design of High Accuracy Nanopore-Based DNA Biosensing. I am delighted to share that this project has recently been approved under the ANRF Prime Minister Early Career Research Grant 2026 scheme, and I would like to sincerely thank the ANRF for supporting this research. The primary objective of this project is to develop a comprehensive mathematical and computational framework for nanopore-based DNA biosensing. With that introduction, let me first give a brief overview of DNA sequencing and explain why nanopore sequencing has become such an exciting area of research. So DNA sequencing is the process of determining the exact order of four nucleotides such as adenine, thymine, cytosine, and guanine that make up a DNA molecule. Now, knowing the sequence is fundamental to modern biology and medicine. It helps us understanding genetic disorders, identify disease-causing mutations, develop personalized therapies, detect pathogens, and study evolution. So DNA sequencing technology has undergone remarkable progress over the past 5 decades, where it began with the Sanger sequencing method in late 1970s and followed by several next generation sequencing technologies such as Illumina or Iron Torrent, which are nowadays also being widely used. However, all of these existing technologies are quite good and accurate, however, they are quite costly. So, on the other hand, the latest advancement in nanopore sequencing, which offers several unique advantages, it enables long-read sequencing, produces data in real time, and requires relatively compact instrumentation. So because of these advantages, nanopore sequences is becoming increasingly important in both the research laboratories as well as in clinical diagnostics. So, before discussing our project, let me briefly explain the basic principle of nanopore sequencing. So we'll start with what is a nanopore. So nanopore is a simple tiny pore drilled at the center of an impermeable membrane which separates two reservoirs, as you can see here. And when an electrical voltage is applied across the pore, ion flow through it which produce an electric current. Now imagine a DNA molecule moving through this nanopore. As each nucleotide passes through the pore that fluctuates, so for example here it partially blocks the current. So there is a fluctuation in the electrical signal. So since each nucleotide perturbs the current differently, the measured current contains information about DNA sequencing. For example, here this one is bigger in size, so the reduction in the current signals is higher as compared to the previous one. By continuously recording this current fluctuation, so one can recognize the nucleotide exactly. So here as you can see, so as the different nucleotide passes by, we have different fluctuation in the current by which one can recognize the exact sequence of the nucleotide. So although this principle is quite straightforward, the underlying physics is extremely complex. So the DNA transport is governed by the combination of electrostatic, ion transport, fluid flow, hydrodynamics, interactions with the nanopore surface. So even small changes in nanopore geometry or electrolyte concentration or material property can significantly influence the measured signals. So the motivation behind this project is to understand these processes qualitatively as well as quantitatively and develop a predictive mathematical model that can guide the design of more accurate nanopore sequencing devices. So the primary objective here is to develop, of course, a comprehensive mathematical model framework for nanopore DNA sequencing. And to achieve this, the project has been divided into 3 levels. So first level, we can focus on the modeling of electromagnetic transport by studying or modeling the Poisson-Nernst-Blanc equation along with the Navier-Stokes equation. And the second level incorporates the biomolecular translocation using the arbitrary Eulerian, Lagrangian or Cayley moving mesh framework. Initially, we will study the motion of rigid spherical particle maybe, and then we can gradually extend the framework towards the object which better represent the DNA. And finally, the third level introduces the fluid structure interaction, allowing us to model the deformable DNA molecule instead of idealized rigid particles. So once the computational framework established, that can be validated against the theoretical and existing experimental data, and the validated model then can be used to optimize nanopore geometry, electrolyte conditions, and material properties to improve the security accuracy. Now, so in level 1 or the first level, the primary focus on the electromagnetic transport inside the nanopore. So here we solve the fully coupled system consisting of Navier-Stokes equation for fluid motion, the Poisson equation for electric potential, and the Nernst-Planck equation for ionic transport. And these equations are strongly interconnected, and the multiphysics formulation forms the foundation of the entire project because it governs the ionic current that is ultimately measured during the DNA sequencing. And to solve this coupled system, we can use COMSOL multiphysics. Now, in the level 2, once the electrokinetic framework has been established, the next step is to model the translocation of the biomolecule. So initially, the DNA can be represented or thought as a rigid particle, maybe a spherical particle, as you can see here. And the particle moves through the board, it experiences both electrical as well as hydrodynamic force. And since the computational domain changes continuously during this motion, the mesh must be deformed accordingly. And this approach enables accurate prediction of particle trajectories and corresponding changes in ionic current during the translocation. And now, in reality, DLE is of course not a rigid particle. It's highly flexible and continuously deforms while passing through the nanopore. So capturing this deformation is essential because it directly affects the ionic current signatures and therefore the sequencing accuracy. So in the final stage of this project, we want to develop a fully coupled fluid structure interaction model that treats DNA as an elastic body interacting dynamically with surrounding electrolyte, and COMSOL is particularly suitable for the task because it provides robust multiphysics coupling, efficient handling of complex moving geometry, and advanced numerical solvers within a unified simulation environment. Now, this project is expected to generate several important scientific and technological outcomes. First of all, it will establish a comprehensive mathematical framework for nanopore sequencing that can serve as a predictive tool for future research. Secondly, it will provide a quantitative design guideline for developing high accuracy nanopore biosensor by optimizing the nanopore geometry, electrolyte composition, and material properties. Third, the predictive simulation will significantly reduce the need for repeated experimental trial and error, thereby reducing both development time and cost. Finally, the project has a strong potential to generate high-impact scientific publication as well as interdisciplinary collaboration and future technology transfer towards the next generation sequencing platform. So with that, I will conclude my presentation, and before I finish, I would like to mention that we currently have an open position for a project associate 1 on this project, and the candidate having background in applied mathematics or mechanical engineering with a prior knowledge of basic fluid dynamics and familiarity with COMSOL multiphysics would be highly desirable. So if you're interested, you can reach out to me. Thank you.